Welcome to Lecture 5. In this lecture, we will talk about three different topics. The first topic involves an unusual oxidation of fatty acids, that is omega oxidation. What does it mean? We'll look at it later. The second topic involves oxidation of branched chain fatty acids. And we'll look at one specific example of a branched chain fatty acids and how it is relevant and why you have to go through a specific oxidation pathway. In the third topic, we'll look at ketone bodies, how ketone bodies are produced and how they are stored. In addition, ketone body regulation in specific disease states. The mitochondrial beta oxidation is by far the most important catabolic fate for fatty acids in animal cells. In addition to beta oxidation, another oxidation pathway exists in animal cells, and this is called as omega oxidation of fatty acids. Unlike beta oxidation, omega oxidation of fatty acid starts from the last carbon. Now, as the name suggests, omega oxidation means the oxidation at the last carbon. Omega, as you may know, is the last letter in the Greek alphabets, right? So the major difference is that the beta oxidation happens or starts from the carboxy end of the fatty acyl CoA, whereas the omega oxidation starts from the last carbon. In mammals, omega oxidation is normally a minor pathway for fatty acid degradation and takes place in the endoplasmic reticulum, right? So this is basically a backup pathway. Omega oxidation takes effect and is important during carnitine deficiency. Now, you all know as to why carnitine is important, right? Carnitine is important because carnitine helps in transporting fatty acids across the mitochondrial membrane. Remember the carnitine transporter? Now, if there is a mutation or if there is deficiency of carnitine, this omega oxidation pathway takes effect and as a result can make smaller fatty acids that can enter mitochondria. Now, this omega oxidation involves three enzymes. It has an oxidase and two dehydrogenase. Let's look at what these enzymes do. The first step introduces a hydroxyl group onto the omega carbon, as shown here. The oxygen for this group comes from molecular oxygen in a complex reaction that involves cytochrome P450 and the electron donor NADPH. NADPH gets converted to NADP+ in this reaction. Reactions of this type are catalyzed by mixed function oxygenases. The product of this first step is then acted upon by another enzyme, alcohol dehydrogenase, which converts this alcohol into an aldehyde functionality. This enzyme uses NAD+ as a cofactor and converts it to NADH. The product aldehyde is then acted upon by another enzyme, aldehyde dehydrogenase, which converts this aldehyde functionality into a carboxylate. In this process, aldehyde dehydrogenase uses NAD+ as a cofactor and then converts it to NADH. It uses a water molecule to introduce an extra oxygen onto this carboxylate molecule. The 12-carbon dicarboxylate molecule that results from the aldehyde dehydrogenase reaction can now get attached to a coenzyme A molecule on either of these carboxylate groups. This can then enter the mitochondrion to undergo beta oxidation. Remember, a fatty acid molecule that is less than 14 carbon atoms can enter the mitochondrion without the help of carnitine or carnitine acyltransferase. Once it undergoes beta oxidation, it results in the formation of acetyl-CoA and other dicarboxylic acids of varying lengths. The length could depend on what the original length of the molecule that enters the mitochondrion is. Succinate, one of the product that is a 4-carbon dicarboxylate, can enter the citric acid cycle and undergo oxidation as a fuel. Branched fatty acids can undergo another type of oxidation called as alpha oxidation, and this can happen in peroxisomes. A classic example of a branched fatty acid is phytanic acid. It's a long-chain fatty acid with methyl branches. It is derived from the phytol side chain of chlorophyll. If you look closely at this molecule, it has a methyl group on the beta carbon. And a methyl group on the beta carbon of a fatty acid can prevent the formation of a beta keto intermediate. Remember, in the beta oxidation pathway, there is a beta keto intermediate, which is then acted upon by a thiolase. So, phytanic acid can no longer enter beta oxidation pathway because of this beta methyl group on the beta carbon, and hence it has to be oxidized. Now, why phytanic acids? Because humans can obtain this molecule in the diet primarily from dairy products and from other fats of ruminant animals. So phytanic acid needs to be metabolized, and it happens in peroxisomes, and alpha oxidation is the pathway that helps in converting phytanic acid into another molecule called as pristanic acid. So what happens here is, in this process, one carbon gets removed. If you look closely, the beta carbon, which has a methyl group, is now transferred to the alpha carbon. And in this process, one carbon gets removed. So how does this happen? You do not have to remember in detail as to what the enzymes and the mechanisms involved in this. You just need to understand what's happening. The first step involves the formation of phytanyl-CoA, catalyzed by phytanyl-CoA synthetase. The second step involves hydroxylation of the alpha carbon. This is catalyzed by phytanyl-CoA hydroxylase. And the third step is when a formyl-CoA molecule gets released from this alpha hydroxy phytanyl-CoA. And the formyl-CoA then gets converted to formic acid and eventually to carbon dioxide. This is catalyzed by the enzyme alpha hydroxy phytanyl-CoA lyase. As the name suggests, there is a lysis that happens. Now this results in the formation of an aldehyde molecule called as prostanal. This is then acted upon by another enzyme called as aldehyde dehydrogenase. We have seen this enzyme before. What this does is it converts the aldehyde into a carboxylate. Prostanic acid, the product, now does not have a methyl group on its beta carbon, and hence it can enter beta oxidation. Beta oxidation of this molecule can result in propionyl-CoA and various acetyl-CoA molecules. Again, to clarify, prostanic acid enters beta oxidation, and the product of the first step is shown here. Propionyl-CoA comes out because there are 3 carbons that is shown here in shaded orange, and the other product is 4,8,12 trimethyl tridecanoyl-CoA. Now, it is important to remember that this molecule can enter beta oxidation again and undergo successive oxidation to release acetyl-CoA and propionyl-CoA wherever there is a branched methyl carbon. One of the fates of acetyl-CoA formed in the liver during oxidation of fatty acids is to enter the citric acid cycle. When oxaloacetate concentration is depleted, acetyl-CoA can no longer participate in the citric acid cycle, and it undergoes conversion to what is called as ketone bodies. The structure of three compounds shown here are called as ketone bodies. Among these three molecules, acetone is produced in smaller quantities, and usually it is exhaled out. The other two molecules, acetoacetate and D-beta hydroxybutyrate, are usually transported by the blood to tissues other than the liver, where they are converted to acetyl-CoA and oxidized in the citric acid cycle. Now, it is important to remember that the production and export of ketone bodies from the liver to other tissues allows continued oxidation of fatty acids in the liver when acetyl-CoA is not being oxidized in the citric acid cycle. The first step in the formation of acetoacetate occurs in the liver, and it is an enzymatic condensation of two acetyl-CoA molecules which is catalyzed by the enzyme thiolase. Remember thiolase from beta oxidation pathway? Now this reaction that is catalyzed by thiolase is exactly the reversal of the last step of the beta oxidation. And in this step shown here, the product is acetoacetyl-CoA. Acetoacetyl-CoA gets converted to beta-hydroxy-beta-methyl glutaryl-CoA by the enzyme HMG-CoA synthetase. And what it does is it adds another molecule of acetyl-CoA to the substrate acetoacetyl-CoA. HMG-CoA gets converted to acetoacetate by the action of the enzyme HMG-CoA lyase, and in this process a molecule of acetyl-CoA is released. Once acetoacetate is formed, it gets converted to acetone by the action of acetoacetate decarboxylase and to D-beta hydroxybutyrate by the action of D-beta hydroxybutyrate dehydrogenase. Acetone is removed as a gas, as I said before, and exhaled, but acetoacetate and beta hydroxybutyrate can traffic to the brain for use in energy production. Ketone bodies are used as fuels in all tissues except liver. The molecule D-beta hydroxybutyrate synthesized in the liver passes into the blood and thus to other tissues, where it is converted in three steps to acetyl-CoA. In the first step, this molecule D-beta hydroxybutyrate is oxidized to acetoacetate by the action of the enzyme D-beta hydroxybutyrate dehydrogenase. Acetoacetate is then converted to acetoacetyl-CoA by the enzyme beta-ketoacyl-CoA transferase. And this enzyme transfers a CoA from succinyl-CoA to acetoacetate. The product acetoacetyl-CoA is then broken down to two acetyl-CoA molecules by the enzyme thiolase. It is important to remember that liver lacks beta-ketoacyl-CoA transferase, and this is the reason why liver cannot use ketone bodies as fuels. In addition, liver contains only a limited amount of coenzyme A, and most of it is tied up in the molecule acetyl-CoA. The production and export of ketone bodies thus frees coenzyme A, and this allows for the continued beta oxidation pathway to take place in the liver. Ketone bodies are overproduced in conditions such as diabetes and during starvation. How does this happen? let us consider what happens during starvation. During starvation, the process known as gluconeogenesis is upregulated. This means that the citric acid cycle is downregulated and the intermediates in the citric acid cycle are diverted towards making glucose, hence gluconeogenesis. As a result, acetyl-CoA cannot enter citric acid cycle and is diverted towards making ketone bodies. What happens during diabetes? As you all know, in untreated diabetes, when the insulin level is insufficient, extrahepatic tissues or tissues other than the liver tissues cannot take up glucose efficiently from the blood, either for fuel or for conversion to fat. Now, under these conditions, level of methanyl-CoA falls. Now, we have seen this pathway before, right? Now, when there is insulin receptors and when insulin is acting through this pathway during high glucose level in the blood, ACC or acetyl-CoA carboxylase is usually activated to form more and more methanyl-CoA. And when more and more methanyl-CoA forms, it inhibits fatty acid oxidation. But in this case, when there is insulin resistance or when there is less amount of insulin due to diabetes, ACC is usually phosphorylated. And hence, acetyl-CoA is not converted to methanyl-CoA. So, methanyl-CoA levels drop. And hence, inhibition of carnitine acyltransferase 1 is relieved and fatty acids enter mitochondria to be degraded to acetyl-CoA, which cannot pass through the citric acid cycle. Again, more and more acetyl-CoA is formed, and these intermediates are drawn off to make ketone bodies. So, ketone bodies are formed during starvation, and ketone bodies are also formed during conditions such as diabetes. Now, this is important. The resulting accumulation of acetyl-CoA, as I said, results in the formation of ketone bodies. The increased blood levels of acetoacetate and D-beta hydroxybutyrate lowers the pH of the blood. So the blood pH is lowered because of increased concentration of ketone bodies. This results in a condition called as acidosis. Extreme acidosis can lead to coma and, in some cases, death. Ketone bodies in the blood and urine of individuals with untreated diabetes can reach extraordinary levels. So in this case, it is called as ketosis, or when combined with acidosis, it can cause ketoacidosis. It's a very serious condition. Individuals on very low calorie diets using the fats stored in adipose tissues as their major energy source also tend to have increased levels of ketone bodies in their blood and urine. These levels must be monitored to avoid the dangers of ketoacidosis. So, let's summarize this chapter. This slide will give you a very brief summary of all the 5 lectures that we have seen for this chapter. So, what did we learn in this chapter? We learned that fats are an important energy source in animals. 2 carbon units in fatty acids are oxidized in a 4-step beta oxidation process into acetyl-CoA, which enters citric acid cycle to make NADH and FADH2. In addition, beta oxidation yields NADH and FADH2 as well. And we all know that NADH and FADH2 can enter the electron transport chain to yield ATP. Also, Acetyl-CoA formed in the liver can be either oxidized via the citric acid cycle or converted to ketone bodies that serve as fuels for other tissues.